My name is Ray Comenzo. I'm a hematologist from Tufts Medical Center in Boston, and I've been working in the field of amyloidosis for the past 30 years as part of an effort in developing new therapies for plasma cell diseases. The work we presented here involves a question, and the question is, why do amyloid deposits accumulate in the bone marrow of about 20% of patients with light chain amyloid? Light chain amyloid is a deposition disease, and light chains produced by abnormal plasma cells aggregate and form fibrils and deposits in a variety of organs in the body. But only 20% of the time do we find amyloid deposits in the bone marrow. This problem has not been examined historically. So we began by sorting marrows that had amyloid and those that didn't have amyloid into two groups. We obtained the marrow microenvironment cells that were not plasma cells from both groups and compared the two groups in a variety of ways, using whole exome sequencing as well as single-cell RNA sequencing. And what we've learned is this: there is a difference in the way the marrow microenvironment responds to the deposits of amyloid. The difference is most apparent in the expression of genes involving red blood cells. Interestingly enough, one of the most overexpressed genes in the marrows that tolerate amyloid is a gene from red blood cells that is also associated with Alzheimer's disease. So we're actively investigating the role of the protein is called TMCC2, in the process of deposition of fibrils. We've also learned that the different cell types in marrows that contain amyloid have fewer hematopoietic progenitor cells, which we think is very interesting. And we're trying to understand what signals are involved in the reduction in essentially granulocyte, monocyte progenitor colonies and cells. And finally, the overall evaluation of cytokine pathways has led us to see that tumor necrosis factor alpha, TNF-alpha, is actively involved in numerous cell types in the marrow microenvironment. And the TNF-R2 receptor is probably the mediator of that. And we're actively interested in the impact of that on the immune system, particularly on T cells and monocytes, because you would expect that monocytes would phagocytize amyloid deposits, but they don't. So we're hoping as a result of this work to get a better insight into the ways that tissues and organs respond to the deposits. Hopefully there will be discoveries that can be converted into therapeutic opportunities.